Hey everyone, it's Mr. Bennett. It is cell activity part 2, and we're going to move now into some detail about how the cell maintains its self, really. So we're going to focus on the plasma membrane. So the big question with this, how do you separate inside from the outside but don't starve? So in other words, how do I keep me as me and other stuff as other stuff, but bring in what I need and get rid of what I don't need? And the answer to that is this thing called the plasma membrane or the cell membrane. It's that covering around the outside of a cell. It's flexible. Animal cells have this. Plant cells also have it, but it's inside the cell wall. But that's the big thing. This is the gatekeeper, right? It's flexible. It can move. It can adapt. It can adjust its size, its shape. We're going to talk about how it does that in just a minute. It also has identifiers that helps the body know what is what. So your cells are unique to you, and this is where we get into questions about transplant acceptance, right? So if I'm trying to donate some organs or donate some blood, I can only give it to people who can accept that. And it's because of these identifiers on our cell membranes. And it also has to do with intercellular signals. So we're going to talk about the cell signaling pathway in a couple of videos, but the plasma membrane of the cell plays a very, very large role in that. So the big question is, well, what does this thing look like? All right, so you can see my big picture here. We're going to come back to that in a minute. We're going to start over here with this. This is the main component of the cell membrane, and it is called a phospholipid. Phospholipid. You've probably seen that word before, but a phospholipid has got two parts. It has a phosphate-containing region called the hydrophilic head. We'll come back to that in just a minute. And the hydrophobic tails are made of fatty acids, right, which are components of lipids. The lipid portion of this is on the inside of the cell, and it's because, if you think back to our biochem stuff, fatty acids are typically nonpolar. All of the atoms in the chain are spread out evenly. There's no pull of electrons one way or the other. There's no oxygens or sulfurs or anything like that is unevenly spreading those electrons out. So these guys are nonpolar. On the other hand, the phosphate group in the head of the phospholipid is very, very polar. It's negatively charged. This phosphate is negative, giving it a polar quality. Because that's negative, it interacts really well with water, because water is also polar. Remember, like mixes with like. So water is polar, so a polar region on a cell membrane is going to interact with water very, very well. A nonpolar region is not going to interact with water. This would be kind of like water and oil, right? So oils are nonpolar, they don't mix with water. The glycerol is still the backbone, so we have a 3-carbon chain in the middle here. So here's carbon 1, 2, and 3. Okay, it forms the backbone, and we get a diester bond across the oxygen that attaches our fatty acid. So it's very similar to just a regular lipid, right, a triglyceride. But instead of a fatty acid on carbon number 3, we have this phosphate group, a PO4 molecule. And that PO4 has a negative charge, and that's where the negative component of this. So phospholipids are the main component in the cell membrane. Because it's got a hydrophilic, a water-loving side, and a hydrophobic, a water-hating side, this is called amphi. A-M-P-H-I, philic. Amphiphilic just means it has two characteristics. So one side of it likes the water, it's hydrophilic. The other side likes not water or likes nonpolar areas, so it's hydrophobic. Amphiphilic just says that this particular molecule has got two sides to it, right? A water-loving side and a water-hating side. So again, phospholipids, we'll talk about this unsaturated fatty acid, right? So there's a double bond there. We'll come back to that in another day. I'm not going to really dive into it today, but this is also important in terms of maintaining the integrity of the cell, especially in cold temperatures. But this is our general structure. So now let's take a look at our cell membrane in detail. I'm going to be moving this around as we go. So the cell membrane, the model is called fluid mosaic. I got to find some space to write this. So this is a fluid mosaic. And doing some early electron micrographs, so very, very low resolution microscopy back in the early 1900s, kind of, actually in the mid-1900s, right around the time that DNA was being kind of defined, it was using the same technique. We could see that there were two layers to this cell membrane, and so the guys working on it, they said, oh, maybe it's got this and that, so through inductive reasoning or deductive reasoning, looking at evidence and then drawing inferences, we came up with this fluid mosaic model that has since been confirmed. So you can see our our cell membrane phospholipids, right? So it is the significant component of the cell membrane. So there's all these phospholipids squished right in next to each other. So here's the hydrophilic head and the hydrophobic tails. And so you can see those in the diagram here, but there's all this other stuff going on. So just to orient ourselves, this is the outside, right, the extracellular fluid, and then this is the inside. Here's our cytoplasm, so this is internal. And that membrane forms the barrier between the internal parts of the cell and the external parts of the cell. This fluid mosaic model is important because if the cell needs to change its shape or if it needs to adapt to its environment, these phospholipids, they can kind of slide past each other, kind of like marbles in a tray. They're not totally freeform, and it's actually a little bit more rigid than you would expect. I could puncture this and destroy the cell. But they have the ability to slide around past each other. So it's fluid in the sense that the cell can change its shape to do what it needs to do. It allows the cell to grow as well. There's all these other components to the plasma membrane. So the large blue objects, right? So here's one right here, and I got to find a pen. Let me change this to yellow. That might help. All right. So here's a protein. We've got a transmembrane protein right here. And then there's some other surface level proteins, like here's a surface protein on the inside. So we're talking about proteins. These are the second main component of the plasma membrane. There are three kinds of proteins, right? So there's surface proteins. Those are specific to internal versus external. So an internal surface protein is a different function than an external surface protein. There is a peripheral protein, and that peripheral protein, it embeds itself into portions of the membrane. It doesn't go all the way through, but it's involved in one side or the other. So it kind of takes some of this internal space. That's called a peripheral protein. And then we've got the integral proteins, and there are specific classes of these called integrins. But these integral proteins, they span the cell membrane. And remember, when we did our protein structure, proteins fold in very, very specific ways. So all of the folds internal to that protein right there are hydrophilic in nature. Or, excuse me, they're hydrophobic in nature. If they're on the inside of the plasma membrane, we don't want them to be polar because that would interact with our hydrophobic tails there. So that would be a hydrophobic portion of the protein. It can be an alpha helice or it can be some beta sheets that are folded over, but the folding is very important. On the external portions then, this is where the hydrophilic portion of that protein would be because it's in contact with an aqueous environment. So these proteins are very, very important. They carry out a number of roles. If you look over on the side, we have a protein channel here. Because this one goes through the plasma membrane, this would be called an integral protein because it transverses the membrane. But this one, its particular shape allows it to open up as a channel and allow things to move in and out of the cell. We're going to come back to these in particular, but these proteins are very, very specific. They're built very, very specifically for a specific purpose and a specific job. So we've got our phospholipids, we've got our proteins. The third component of our cell membrane are sugars, carbohydrates. You can see, let me grab my pen again, on our proteins, on the cell membrane, so looking at this guy right there. So there's a protein sticking out there. It might be an integral, it could be a surface level, we're not quite sure just because of the way it's drawn. But on the outside of this, there's a chain. If you can see that, that's a carbohydrate chain, and that chain is mainly for cell recognition. When these sugars attach to proteins, they're called glycoproteins, right? Glyco meaning sugar. And when they attach to lipids, so here's another one. We've got there's a chain right there of some sugars, some carbohydrates attached straight to the phosphate group. So this is called a glycolipid. And all of these are used for cell recognition. So your body has a specific pattern of glycoproteins and glycolipids that is unique to you. And they're on the exterior of your cell. You can also have some on the inside, right? So there are some proteins that do things. They're sugars they can attach. So here on this alpha helice protein, this alpha helix protein, here's a glycoprotein tail right there. And then there's also one here on the inside. So they're not unique to outside only, but the main component or the main portion or the main reason for the sugars is for cell identification. And finally, the last one, there's the fourth, and you got to look on the inside here. So think back again to your biochem. We've got a 4 carbon chain, or a 4 carbon ring, or a ring of 4 rings of carbon, sorry. So this is cholesterol. It's a lipid. And that is used to help keep the fluidity of the cell membrane. So cholesterol, it's a steroid ring, and it attached to the head, so this is still nonpolar on the inside there, and it attaches to the hydrophilic head. But it helps keep the cell membrane fluid, especially in cold temperatures. And again, we'll come back to that when we do some of the specifics. But you have to recognize that they're also a component of the cell membrane. So again, to recap, we've got four main components of our plasma membrane. The phospholipid, right? The phospholipid is composed of two parts. We have a hydrophilic head that contains a polar phosphate group, a negatively charged phosphate group, and then a hydrophobic tail that are fatty acids attached to a glycerol. And those are the main component of our plasma membrane. Within the plasma membrane, there are some proteins. They can be integrated proteins, transverse in nature. They can be peripheral, which means they're only through one portion of the cell membrane, or they can be surface level, just kind of tagged on the top there. Carbohydrates are tags on the are tags on phospholipids, or on the not the phospholipids, on the glycolipids or on the glycoproteins, and they're used for identification. And then we've got some steroids that fall in the middle there.